share.
That looks like that.
Yeah.
Presentation one.
Yeah, it's showing the right.
Yeah.
Cool. Now we're moving to the talks. We have with us Olivia, and she's gonna talk to us about determining of a drug concentration in Auckland City Air using LC-MS using MRM analysis. Thank you very much for coming.
Thank you. Kia ora, so I'm Olivia. I just submitted my master's, so please, fingers crossed. So I'm going to be telling you a little bit about my project today. So basically, the prevalence of drug use has increased dramatically over the last few years around the world. We're looking at about a 30% increase in drug use in the last 10 years alone. So when drugs are handled or used, this can lead to small amounts of that drug being emitted into the air. So this could be when you're crushing or handling a drug powder, or if you're heating a drug before use. However, when you're smoking drugs, this is likely to contribute the greatest amount into the air. So in New Zealand, some drugs that are commonly smoked include nicotine, cannabis, and methamphetamine. So as you can see in the picture here, that cigarette smoke actually contains lots of aerosol and particulate matter. So particulate matter is basically lots of tiny little particles that become suspended in the air. They can range from quite big sizes at hundreds of microns, all the way down to just nanometer scale. And it can include a wide variety of things, such as pollen, spores, dust, combustion products as well, from your home heating, your vehicle emissions, or smoking that cigarette on your lunch break. So a lot of things to contribute there. So we're talking about detecting drugs in the air. We're looking at really, really, really low concentrations, which in itself I think is really exciting. If we can collect data on what kind of drugs are present and what concentrations in the air in New Zealand, we can compare these to levels in other countries, where similar studies have been completed recently, such as Spain, Algeria, and Italy. Air sampling for drugs also provides a complementary drug monitoring method to wastewater analysis, which is something that ESR currently does in New Zealand. I'm sure you all heard about the COVID-19 wastewater testing, kind of the same principle, but for drugs. We can then use that kind of information to investigate areas of drug use for air sampling, rather than areas of drug excretion, which is what wastewater looks at. And then this kind of information could be used to inform harm reduction strategies around the country. We could also use information on nicotine in the air, for example, to inform our smoke-free 2025 goals. Because as we've seen again from COVID-19, the public does really enjoy a little bit of scientific information if you can give it in that kind of bite-sized number. So basically the overall project aim was to develop a method to determine drug concentrations in the air and to carry out the sampling to do that. So for my project, it was only a year long, I chose 4 analytes of interest, and you can see the chemical structures on the slide. So that's nicotine, methamphetamine, caffeine, and tetrahydrocannabinol, or THC, which come from cannabis. So in order to collect my air samples, I had this air sampling pump, which basically sucks a whole lot of air through a Teflon filter during the sampling process. So you can see here by this arrow that the airflow is going into the inlet, which is where the filter is housed. I then had the whole pump system in this weatherproof housing, which is this box you can see going on top, because it was in the rain at the sampling site. So the main sampling site where I sampled was at Customs Street West. You might go past it on the bus in the morning. It's an air sampling location that the Auckland Council currently uses. So I sampled there for about 5 weeks, from Monday the 7th of December through to Monday 11th of January. And over that time, I collected 10 samples. So 5 representing weekdays and 5 representing weekends. So the weekday samples, they were collected for about 96 hours from a Monday mid-morning through to Friday mid-morning. And then the weekend samples from Friday mid-morning back around to Monday. I also collected a sample at a New Zealand music festival for some comparison, which we'll talk about later. So this map just really quickly shows you the orange dot is where the sampling site was. You can see the zoomed in picture at the top. It's basically just a cage with a whole lot of really expensive equipment inside. So I set up my pump there, and as you can see, it would have got rained on. This picture is a little bit more interesting. These are some of the filters from the process. So on the left is a blank unused filter. It's clean, it's pristine, straight out of the box. And then in the middle is a filter that sat at Custom Street for 4 days, sampling the air that we breathe down there. On the far right, you can see the underside of that filter, which really just shows you how much particulate matter was collected on the filter while the air passed through. So think about that in your lungs next time you're walking down there. So then these pictures up here show you what the sample extracts look like. You'll notice that they're quite opaque, some more so than others, and that's because of the amount of particulate matter that came off of the filters. Some obviously had more matter than other filters. So darker or lighter colours in the extracts. To do the extraction, I used a non-specific method to see what was there, and this was based off a method used for methamphetamine wipes already in the group. So each of the filters were cut in half to retain half of the filter for other kinds of analysis, and then they were spiked, the half for extraction was spiked with methamphetamine D9 as an internal standard before just being soaked in methanol for 5 minutes, vortex mixed for 30 seconds, and then squashed 3 times with a syringe each. So that's what you get, this dirty looking watery kind of sample or methanol sample. And then I filtered each of the extracts because I know I would have gotten a lot of trouble with our technician if I put these straight on the LC. So yeah, definitely filter those. I also calculated the extraction efficiency for all 4 of the analytes. And as you can see on the slide, it's pretty decent. So I wanted to use MRM with LC-MS for my analysis. And in order to do this, I had to develop an LC method that was going to separate all of the analytes from the sample. To be honest with you, this took me more in the months kind of category to do, despite there being a few other methods out there already. But in the end, I ended up using a biphenyl 50 ml column, which is a column that's designed for the separation of drug compounds and other similar molecules. So that was a lot better than the C18 or PFP columns I had tried. I used a mobile phase of water with 2 millimolar of ammonium formate and formic acid, and then methanol. And these were combined in a gradient over a 12-minute runtime. So I feel like that's relatively short. And this is what I got. So this is all 4 of the analytes, well separated, achieving complete baseline resolution. So I was pretty happy after a couple of months to see this in the data. So I know that most people here have probably heard about MRM before, but just to make sure everyone's on the same page, I'm going to go through it really briefly. So MRM, or multiple reaction monitoring, is a form of targeted analysis performed using a tandem mass spec. In my research, I used a triple quad with electrospray ionization in positive mode for all of my analyses. So basically what happens in MRM and what I did in my project is you have to select for a parent ion in the first quad. Okay, so in ESI in positive mode, the parent ion is typically 1 mass to charge unit above the molar mass of your compound, so your M plus H ion. Then in the second quad, you're applying more energy onto that parent ion in order to induce fragmentation. In your third and final quad, you'll select for ideally at least 2 daughter ions to really get to the end there. You select for those 2 daughter ions and it's the response for those that is recorded. So I said you should select for 2 daughter ions, and that's because you want a quantifier ion, which is used to determine the amount of sample you have, and then a qualifier ion, which really confirms the structure, making sure that what you've got is actually what it is. So there are a few benefits to MRM, but the main benefit is that it gives you increased selectivity and increased sensitivity by avoiding any of those background levels that you might see in your sample. And as you can see from the filter that I showed a couple of slides back, I'm sure there are a lot of background things in that sample other than drug compounds. So once you've selected for your parent ion, your M plus H ion, you need to carry out a product ion scan. Okay, so a product ion scan is basically where you select for your parent ion in the first quad, apply energy in the second, and then complete a full scan of all of the daughter ions for all of the fragments that happen as a result. You can then use the abundances and the mass spec of each of these ions to select the daughter ion for your compound. Now, ideally, it would be great to select daughter ions from both sides of a molecule, so you kind of get the whole structure, but based on the abundancy of what ends up there, it's not always possible. So why would you bother doing MRM? Well, the answer is the limit of detection is awesome. I tried 4 different analysis techniques during my project. I guess you could call them 4 too, you know. And you can see that the numbers in the bottom row are by at least 2 orders of magnitude better for each of the analytes than the other techniques. So these limit of detections are reported in picograms per cubic meter. So I've done the calculation based on the average volume of air that I was collecting in my samples, which was 358,000 litres. So, you know, pretty big sample here. These are in the parts per quadrillion range, or ppq, which is your 10 to the negative 15 zone. So just to kind of give you a bit of perspective, because this is really, really, really small, that's equivalent to 1 drop in 20,000 Olympic-sized swimming pools. So anyone watching the Olympics, imagine 20,000 of them and then 1 drop. So I want to go through a little bit of an example just to show you some of the method development I did for methamphetamine. So this is a total iron chromatogram of a mixed drug standard, just containing those 4 analytes. And I'm going to be honest, it's pretty gross looking. There's a lot of peaks, it's kind of nasty. So one of the things we can do to improve that is to take an extracted iron chromatogram, just looking for 150 mass to charge units, which is the parent iron for methamphetamine, because methamphetamine has a molar mass of 149 and it's singularly charged, where ESI positive mode. So that gives us those 2 peaks in green, as you can see, but we need to work out which one's methamphetamine and what daughter ions we're going to use. So the next step then would be to carry out a product ion scan. You can see it fundamentally looks the same as the EIC. So we're going to take a look at the mass spec of both of these peaks. So we'll call the first one the peak at kind of 3 minutes, and the second one the peak at about 9 minutes. So on the left, you have the mass spectra for the 3-minute peak. Okay, you can see that there is quite a prominent ion at 91 and another at 119. Based on the literature available, these are very characteristic fragments for methamphetamine, so we know that that's what this peak is. You can see that these would also be really good daughter ions to select because of the abundancy in the sample, but they're not kind of from both sides of the molecule. Oh well, you know, doing the best we can here. So we would select 91 and 119 for methamphetamine. And just to confirm, if you look at the second mass spec for the 9-minute peak, you'll see that none of those ions are present, and so therefore it's not methamphetamine and likely some other contaminant in the sample going on there. So then this is the final MRM that you would get. So going from that TAC you saw at the start to the MRM at the end, you can see that this method is really worth it. All 4 of my drugs that I selected are here, and you'll see that the black peaks are the quantifier ion in the highest abundancy, and then the red peaks are the qualifier. So for methamphetamine, which is the second peak to alert, the black peak is the 91 ion, and the red is the 119. You'll also notice that that little peak around 9 minutes that would come out just before THC is gone, and that's because we've got rid of all of the background noise in the sample. So if that was a standard, you can imagine just how much was present in the samples. So I use this method then to look at the filters I collected at custom stream. So nicotine and caffeine, no surprises there, were present at custom stream, all 10 of the samples that I collected. Nicotine was present at an average concentration of about 6,500 picograms per cubic meter, while caffeine was present in a little lower concentration at 27 picograms per cubic meter. So both of those are widely consumed drugs in New Zealand. Both of those are also perfectly legal, so don't stress about that coffee you enjoyed at lunch. But we know that about 73% of New Zealanders consume caffeinated beverages, while only 13.4% of New Zealanders are smoking tobacco. So it really goes to show that that method of drug administration has a huge role in the airborne drug concentration, because a whole lot less people are smoking tobacco, but we're seeing a whole lot more nicotine in the air. So while there's not really any countries that have done similar analyses that are particularly comparable to New Zealand, I kind of thought that the data from Algeria versus the data that I collected was quite interesting. So in New Zealand, cigarettes are really expensive. We have one of the highest prices of cigarettes in the world. So it's no surprise that we have much higher prices than Algeria, where you can pick up a 20-pack of pre-rolled cigarettes for the equivalent of New Zealand $2.80. Whereas the same pack here would set you back at about $37. So quite different. Algeria also has really different restrictions on tobacco, particularly on smoking in public. So there's no rules around that there. And they also have really different restrictions on the sale of tobacco. So in New Zealand, you're not allowed to smoke in public places, outside schools, all of those kind of things. But that's all good there. So it's really no surprise, I guess, that the concentrations of nicotine detected in their study in Algeria were 19 times higher than what we saw in the air here. So I also investigated the correlations between nitrogen dioxide and caffeine and nicotine concentrations. So I looked at nitrogen dioxide from nitrogen dioxide commonly comes from bus exhaust as one of the big contributors. And so some people would use it in research to be a proxy towards activity. So we're going to use NO2 levels as a proxy to activity in the CBD. You can see in the graph on the left that nicotine concentrations are in blue over the 10 samples that I collected, and caffeine concentrations in the graph on the right are in green. The grey line on both of the graphs is the nitrogen dioxide levels detected for those sampling periods, so the weekdays, weekends, and you can see that the trends match up relatively well. So the trend of nitrogen dioxide is quite similar to both caffeine and nicotine, suggesting that days where there's lots of activity or times where there's lots of activity, there's likely to be higher concentrations of nicotine and caffeine. So you can see that sample 3 is particularly high. That's just before Christmas. So you can imagine the CBD was pretty busy around then. I can personally guarantee that it was, because I had to go down there every couple of days to collect my samples. So lots of people around on that week. I also see I looked at THC and methamphetamine. So in the custom street samples, 4 of them contained THC and 6 contained small amounts of methamphetamine. Again, these are at much lower concentrations, 4 and 28 picograms per cubic meter respectively. I also collected this sample at the music festival. If you remember, I said that at the start. And that was the highest THC concentration that I detected in the sample during the project. So, you know, kind of not surprising, I guess. And that was nearly at 35 picograms per cubic meter, which is actually similar to a concentration reported in a Spanish study, just in their everyday urban environments. So, you know, I guess the legality and some of the perspectives around cannabis might be a bit different there. So I used that value and I did a calculation based off values reported in another paper. And I determined that the average person is likely to inhale 1 dose over about 16 million years of THC. So I mean, there's a few limitations with this calculation, such as like the proximity you are to the person smoking cannabis or the time that you are in that proximity. But it just goes to show that while this number is really, really small, you can use MRM analysis to detect things in this picograms per parts per quadrillion range. There's 1 drop in 20,000 Olympic swimming pools, which I think is really awesome. So that's me, and a huge thank you to everyone who was involved in my project. Several people are here today, and to the Auckland Council for letting us use their site, and Mod for helping with the sample collection, particularly Hamish.
In Amsterdam.
Yeah, it would be interesting.
That would be very interesting. You go there and you're like, am I smoking? So do we have any questions? A very quick one, perhaps? Yes, sure.
Was your ticket to the music festival a research-related experience?
I can't answer that. I myself didn't actually go to the music festival. I just received the sample after that. Yes. I wish it was a research-related experience, so maybe I could go to all the music festivals next year. Do you have a question?
You mentioned you showed the NO2 at the same time as drugs. Where did you get the NO2?
So NO2 is one of the things that is monitored in the air by Auckland Council. So that data comes from them.
Thank you very much. One quick
Sorry,
question.
Christian, do you ever see the effects of the environment on your readings and stuff in terms of things, wind direction, temperature, and things like that?
Yeah, so I'm glad you asked. That is something I looked into. I collected weather data at a nearby site, which again is collected by Auckland Council. I looked at rainfall, humidity, wind speed, wind direction. And the only thing I found a correlation between was nicotine concentrations or drug concentrations and wind direction. So when the wind was coming off the harbour, the levels were lower, and when the wind was coming from the side of the city area, the levels were higher. But again, that correlation was on the weaker side of things. Yeah.
Cool. Thank you very, very much. All right, we are moving on to our next presentation. We have Melissa Kirk, investigating the defense strategies of the exotic weed, wild parsnip, and using GCMS. Thank you very much for coming.
Thank you.
Just this one. Well, and then-
You can be my
Today my talk is a little bit different to what you've seen today, because it's ecology based, not really chemistry based. We're using chemistry in it. So I was looking at a exotic plant called the wild parsnip, which is found within New Zealand, and it has certain chemicals that it uses for defense to protect itself against herbivores. And so there's 8 key known furans that are found within the flowers of this plant, and that is what I looked at as part of my research. Some background to my research is that I'm looking at rapid evolution of weeds and the interaction between herbivores and weeds in exotic ecosystems. And so weeds can evolve rapidly within the introduced environment in a few different ways. One of them is the EICA, which is the evolution of increased competitive ability. It's the idea that when it's in the exotic ecosystem, it's lost its specialized herbivores, and therefore instead of putting resources into defense, it puts it into growth instead. And then the other one is the shifting defense hypothesis, which is depending on whether it's being fed on by generalists or specialists, can influence the chemical defense protein, the chemical defense composition of the plants. And some chemicals are look more likely to the generalists, but those same chemicals could attract specialists. Okay, sorry. So my case study is the wild parsnip and its natural enemies. So it's got furacumins in the wild parsnip, which basically form resistance against its specialist herbivore, the webworm. And when webworm damage occurs, it inflicts higher response of toxins and bergapin and spondin, and the genotypes of these plants which have increase of these chemicals is that when they're in the absence of the herbivores, because it's possibly it's not as good, whereas vice versa, the other way around. Before the reassociation of webworms in North America, however, there was a reduction in the chemical defense of this plant, but then after 20 years of reassociation with the specialist herbivore, they increased their defense again. So the wild parsnip within New Zealand, it was introduced for food source, and it escaped and became naturalized in 1867. Webworms were first found in New Zealand in 2004, and so the plant enjoyed at least 140 generations free of the webworms. The webworms can cause a very high amount of seed loss in New Zealand populations. And a previous study done in New Zealand on the reassociation of the wild parsnip and webworm found that they had lower defense chemicals compared to the US, and an increase in tolerance. So the plants were actually larger here compared to the US, and larger with increased time since reassociation. We have also found that there are new generalist herbivores feeding on this plant, which goes to support the idea of a loss of chemical defense. So we found 2 different capital species from New Zealand feeding on this plant, but it was at relatively low rates compared to the specialists. So these are the chemicals within it. They have slightly different roles within the plant itself. So octobutyrate is a deterrent of herbivores but attracts pollinators. Octoacetate is an attractant to webworms, so they use it to cube the wild parsnip, and it can attract some pollinators and deter other pollinators. It's dependent on the species. We get a correlation between xanthotoxin and myosin. So when they work together at high levels, they increase the rate of mortality in the webworms. And bergapin and strontium is metabolized more slowly, so they might be key to resistance. And then in New Zealand, parsnips, they've been found to have lower levels of some chemicals and higher levels of others compared to the US. So optimum defense of flowers, this is the idea that as the flowers develop, they increase in their chemical defense. So for wild parsnips, on each umbel, which is a collection of flowers, they go from buds to males to females to then seeds and fruits. And as they increase, it's been previously found in the US that as they develop, the increase in chemical defense exponentially increases as well. So my study, I wanted to look at optimum defense theory in the New Zealand wild parsnip flowers. Does it happen within New Zealand, and does it happen at an increased rate with herbivores versus without? I wanted to look again at tolerance, but more within the New Zealand population. So you have a range of sizes of small to large plants, and whether some are using tolerance versus others are using chemical defense. And then overall, the patterns of the chemicals with and without the herbivores. So I sampled wild parsnips across New Zealand from sites with wild generalists and specialists and with different times since reassociation. I collected information on plant heights and number of rules and infestation rates, and I used GCS MS analysis on the flowers to look at those 8 key fluorocumarins, and then I did some statistical analysis in R to look at the differences between those 8. Okay, so optimum defense within the flowers. So is it happening? Yes, as you go from male flowers to more developed seed flowers, it does increase. Unfortunately, I don't have it in consensus order. The archive was not working. So so it's male flowers, female flowers, hermaphrodites, which is the between, and then seeds. So yeah. But yes, it does increase all but the myosin. So there is evidence that as they develop, they are still increasing their chemical defense as the plant is wanting to protect those more vulnerable and more valuable assets, which is the seed. So optimum defense is an increase in the presence of herbivores. So I looked at it under herbivory in general and under just webworm herbivory, which is a specialist, and it has generally the same pattern. It does not exponentially increase under the presence of herbivores. I also wanted to compare the optimum defense of the flowers with New Zealand and the US, as the US has an increase in the chemical defense. We found generally similar patterns of increase going from the different flower stages. However, in New Zealand, for going from male to the more developed seeds, you have an increase of the spondin and a decrease in the bergaptin, and when you go from males to females, again, you have an increase in bergaptin for the US, but a decrease in New Zealand, an increase in xanthotoxin, and then somewhat similar increases for female flowers to seeds, but slight increase in Spondon and Bridgerton for the US compared to New Zealand. So I also wanted to look at the dual strategy. So we know in New Zealand there's an increase of tolerance. So overall, New Zealand wild parsnips can tolerate herbivory more than the US. However, we wanted to see whether within those populations, when you get smaller plants versus larger plants, are the smaller plants more chemically defended. We did not find that to be the case, and we didn't find it to be the case under herbivory or under specialized herbivory. So it seems as though they are not doing the dual strategy of smaller plants are more chemically defended and larger plants are more tolerated. Instead, we found actually that the height of the plant was correlated with octoacetate and imperatin, and then under herbivory, mycins increase in height as well. We did find, however, smaller plants were less likely to be infested by herbivores. This could be due to smaller plants have a smaller production of flowers, less flowers produced, so less octoacetate would be produced, and less small, and per gram could be smaller since there's an increase in the height. And so therefore, potentially, smaller plants are just more inconspicuous within the environment compared to larger, and therefore avoid herbivory that way rather than chemically. We wanted also to look at the reassociation with herbivores, so they've got different timeframes in terms of when they were infested by the webworm. So older associations did not have increased chemical defense like we expected. Mycin was a little bit higher, but the other compounds were not. And but instead we found that older sites had again increased height, and therefore potentially are shifting their defense to tolerance. We found no associations, however, did have an increase of spondin and also Bridgerton as well, which are the two chemicals that are associated with webworm herbivory. So we wanted to also look at the role of herbivory. This is just a simple yes, no for herbivory. We found that octoacetate was significantly higher in areas with herbivores, whereas octobutates and imperatin were lower. We then looked at it in terms of the generalists versus the specialists, and we found that octobutate was significantly higher for all flower samples from sites with generalists and with no herbivores at all compared to specialists, but octoacetate was higher for sites with specialized herbivory. So this is not what is predicted by what happened in the US and from the natives as well, where xanthotoxin, sporin, and the Bridgerton all normally increase under webworm herbivory. So the conclusions are that it supports the idea that optimum defense strategy is occurring within New Zealand wild parsnips in terms of their flower development. They're still partitioning more of the chemical defense to more developed flowers, but it is not heightened under herbivory. Findings do not support the dual strategy of tolerance of chemical versus tolerance in terms of smaller versus larger plant sizes. And then we did not find that xanthotoxin, sporin, or Bridgerton were greatly increased in the presence of herbivores or even in the presence of the specialists. However, recently they did have an increase of two of these chemicals. Octoacetate was a key chemical that increased with herbivory and particularly specialized herbivory. And its role within it, although it does attract webworms, it also attracts some pollinators. And when wild parsnips are infested, their pollination overall visitation is reduced. So it could be that they are needing to produce more of this to attract those more promiscuous pollinators. And so pollination might be also a key driver in this. Additionally, this octoester is related to the furocoumarins in terms of their ability to penetrate the gut of the webworms and therefore enhance the cumins, and so maybe in New Zealand an increase in this octoacetate is beneficial, but also will be attracting more workers to the plant as well. I still got a lot of analysis to do with this data, so I want to look at the chemical profiles as a whole, not just each individual of the 8 chemicals. And I want to look at the role of flowering times of the last to see whether there's avoidance. And then also I have another data set of a different year to compare with, and potentially look at other chemical defense compounds that might be present as well. Sorry for all their help, because I'm not a chemist. Yes.
Thank you very much. Thank you very much. Do you have any questions? Oh. Yes.
I just wondered about going back to look at other chemical compounds. Are you going to remine the data, or you've got some more sample to do?
I've got basically all the chemicals within it. I just focused on those 8 because they already have known roles for different stuff. But I have to go through and normalize all the data and stuff, so that'll be fun.
And is that something you're going to do sort of yourself with your R, or is your R more on the statistical testing side of things? You're going to
So I normalize it with bringing it through R and originally look for batch effect, and then also biomass and internal standard, and then I analyze it through R in terms of statistical analysis.
Yeah.
She has the full profile with through the GC. She only concentrated on those 8 peaks, but there are hundreds of peaks there. So now we can analyze the full profile and see if there are other interesting chemicals out there.
Thank you. Thank you very much, Melissa. All right. So our next speaker is actually one of our MASH collaborators. Chris, can you hear us? Yes, you can.
Hello, can you hear me?
Chris trained his voice yesterday on the workshop. Thank you for joining us. Yeah, okay, so you can share your screen, right? Yes, awesome, great.
Okay, good. Can you hear me now?
Yeah.
Awesome. Well done, guys. Thank you for going above and beyond to make it possible for me to present to you today. I'm very pleased to be here. Kyriakos Varnava. My name is Chris Pook. I am a research fellow at the Liggins Institute, specialising in analytical chemistry, biochemistry, and mass spectrometry. I'm here today to talk to you about exploring the volatilome using thermal extraction and GCMS, and to tell you about the lessons I've learned from 6 months of method development. So this is the analytical platform you can see on the screen in front of you that I'm using. It's fairly standard thermo GCMS. The trick is the Gestel autosampler on top of the instrument, which has a thermal absorption unit. Can you see my cursor?
Yeah.
Yeah, good, lovely. So you have this thermal absorption unit on the front of the instrument. Now, conventional GC sample introduction techniques rely on the introduction of samples either in liquid form, where the sample needs to be mostly volatile and carried in a volatile solvent, and injected into a hot splitless liner. And that relies on an extraction technique which selects for purely volatile compounds, and it's quite rare. So you typically have to go through some sort of elaborate some elaborate cleanup techniques to remove non-volatile contaminants from your liquid extract. Otherwise, those things, non-volatile compounds, will tend to accumulate in the injection liner and cause you problems which can interfere with the balance, the dynamic equilibrium between metabolites in the solid or liquid phase in your sample, in the gas phase, or absorbed to the fiber. So the Gestalt thermal desorption unit is it's a bit of a racehorse. It's a very cool thing. It comprises two heated zones. On the top is a thermal desorption unit, into which is placed exchangeable liners by the autotumbler. That's the top bit here. And that meets another liner, which is part of the cooled inlet system on the bottom. The cooled inlet system functions both as a cryo trap and as a heated injection port. So samples are added to the TDU by the robot. They get heated to desorb volatile targets. Those are carried on the flow of gas into the cryo trap, but are cryogenically trapped and focused. At the end of trapping, the TDU cools down again to limit release of further volatiles, and the fifth starts to heat ballistically to revolatilize the sample and release it into the GC column for conventional GCMS analysis. Here's a little video of the beast working. The tubes on the left show you the multiple ways in which you can introduce samples into the TDU. You can, if you choose, actually just place solid material in the tubes. You can also insert tubes that are packed with sorbents, and using an offline sample collection system, you can pass gaseous samples through these tubes to collect volatiles in air or headspace samples. Those are then firmly desorbed into the GCMS. The third one here is called a twister, which is a little magnetic stir bar coated with a DCMS sorbent, which is quite similar. The previous one is the sorbent phase, which you trap offline. And finally, you can add little glass microvials into the tube containing liquid or solid samples. When most are heated, the volatiles are oiled out of that sample and carried in through the machine below. Here's an example of the applications that we've developed for this platform over the last few years. I know the title of the talk mentioned 6 months of method development, but I have been working with this technique for more than 3 or 4 years now. So we tried using these sorbent stir bars coated with PDMS or ethylene glycol phase to sample skin volatiles using this little metal holder to roll the stir bar along participant skin. I think this is Nita McKenzie, one of our summer students who's carrying out the analysis, being the guinea pig here. That stir bar then goes into the TDU tube and is placed into the TDU for desorption thermally. Here's an example of a packed tube trap, packed with 10x. And by coupling this to a metering pump like this, you can pass a fixed volume of air through it, and that will provide you with quantitative sampling with reasonable sensitivity in quite a short period of time, and good sensitivity. 10x is a very commonly used sorbent phase, although there are many other sorbents that you can use to customize your application. We've already published some work using the TDU platform from my previous role, where a research fellow at SMHS, Harish Devaraj, worked with me to profile the headspace volatiles from E. coli cultures using these little sorbent stir bars, which we trapped on the lid of a petri dish using magnets to collect the volatiles released from the E. coli cultures. This is an example of the ethylene glycol results. On the top is the inoculated sample. On the bottom is the medium length. So you can see there are a lot of volatiles present in the length. But we've got, because of our good procedural blanks, we managed to find some interesting volatiles. Benzyl alcohol there, phenethylcreel alcohol, picresol, and of course this big one here is indole, which is already well understood to be an abundant bacterial alcohol. We've got slightly more results using a slightly different sorbent phase. human stool samples, and labs. And you can see some of the homogenized stool extract being lyophilized, ready to go into the TDU. And this produces an incredibly rich chromatogram, very diverse set of features. And we're very happy with this result initially. Unfortunately, we saw exactly the same problem that we found with the Kyriakos analysis, in that a lot of the higher boiling compounds were not effectively extracted or quantitatively extracted from the sample. And these are very small samples. The actual sample mass in these tubes is about 1 milligram dry weight. So it's a really tiny sample, but even under some really high temperature extraction conditions, we weren't pulling out these things. And that should be surprising. This cluster of peaks at the right-hand end of the chromatogram are sterols, single sterols. So they have boiling points above 300 degrees. If we plotted the fatty acid profile, because there's actually a very wide range of fatty acids in these stool samples. If you plot boiling points of those fatty acids against their retention time, you can see that these are the longer chain fatty acids at this end. So this is stearic acid, this cluster of dots here, palmitic acid, and their boiling points are above 300 degrees. And that's problematic because the maximum temperature of the TDU is only 350 degrees. So we're reaching the limits of the platform to be able to slowly desorb these things. In fact, we don't want to work up around those temperatures because at the same time as we are volatilizing long-chain fatty acids, unfortunately, we're also starting to release triglycerides and sterile esters, which are very high boiling compounds. And those cause serious problems because those things accumulate in the ejector and hang up really nastily, and you end up with some quite heavy contamination problems. One of the other problems you can see from that plot is that these shorter chain fatty acids are spread across quite a long region. And when you look at chromatograms, extracted ion chromatograms for fragments representative of fatty acids, you can see that at NZ60, 73, and 74, which are characteristic fragments of carboxylic acids. The later peaks are not beautiful, but they're reasonably well resolved. This would be palmitic acid, this would be stearic acid. Whereas down here, the short chain fatty acids are not resolved at all well. So that's problematic because these guys are actually very interesting from the perspective of stool analysis. But the other problems that we found was around carryover blanks. So we analyzed the stool sample, which produced a chromatogram at the top, and then followed that analysis up immediately after with a vial blank with an empty tube and also sample in. We still saw carryover. So this wasn't a problem of unextracted material. This was volatiles which had accumulated inside the DEU and were contaminating it. And we had to undergo an extensive cleaning procedure to get rid of those things. Unfortunately, those things were also accumulating in the ejection liner, and this essentially makes this analysis non-viable. This wasn't the end of the world because we have other analytical techniques accessible to us, for which most of these target compounds are amenable. So that's not too much of a loss, just giving up the analysis of things like sterols or high molecular weight fatty acids. The things that we did want to actually quantify, which are part of the goal of this stool analysis, are the short chain fatty acids. So once we'd resigned ourselves to not being able to collect those high boiling compounds, we could make some more pragmatic decisions about column choice and extraction methods. And we moved to a much shorter method, a different column chemistry, which was much better at resolving short chain fatty acids, which is this kind of data you're looking at here. So these cluster of peaks, this is a standard run at the top of a volatile fatty acid mix. And you can see we're getting very nice resolution of all these short chain fatty acid peaks here. Here's a representative sample from the stool extract. This is acetic acid, this big one here. Proteinolic, methyl proteinolic, and butane. So So this is really like chromatography now. We pursue this work a lot further because this is a seventh goal. So the reason we're looking at short-chain fatty acids is very important for our gut health because they provide an energy source of gut acid and keep it healthy. They stimulate mucus production, which is one of the functions of the gut epithelium to prevent bacterial ingress and to keep barrier between the contents of the colon and the blood on the other side. Short-chain fatty acids are also involved in modulating the immune function, and they're also a possible contributor to obesity. So the feedback is through metabolism of non-digestible carbohydrates, and they are linked to chronic inflammation phenotype as well. These are all things that we were interested in studying. So it's very important to be able to quantify these things accurately. And unfortunately, there's not many choices when it comes to very small molecules like acetic acid and proteinic acid. There are some ways that you can do them orthogamy using liquid chromatography and derivatization, but those are fairly limited, and they don't give us the opportunity to explore the rest of the volatilome, which, as I'm sure you can understand, in the full samples is actually fairly rich. But also might have some very interesting biological functions in terms of modulating host physiology and communication between the microbiome and the host. So the method that we finally settled on resolves 10 different fatty acids, short-chain fatty acids beautifully. Here you can see we have on the left acetic acid and subtly labeled internal standard in red. And then you have carbons 3 to 8, several branch chains denoted by B. The red ones are the internal standards that are subtly labeled internal standards, octanoic, hexanoic, and acetic. And we've now got Kyriakos Varnava. You can see down there, C8 is the standard, by the way. Octanoic acid wasn't actually a part of the standard, but you can still see there's a little blip down there on the baseline, the weekly baseline result being internal standard, and the unsaturated one. We've got linearity using our internal standards. Very good linearity across the range. R squared all those exceeding 0.99 comfortably. The reproducibility of the analysis is pretty good. We're always below 15% RSC from 8 replicate analyses, and definitely below 10% RSC for these targets. I'm not including heptanoic and octanoic in this table because those aren't abundant in food samples, so we're not focusing on those guys. Excuse me. Okay. One interesting phenomenon is the presence of these two short-chain fatty acids, 2 and 3 methyl-butanoic acid. These are actually quite interesting. They're products of branched chain amino acid metabolism, and they've been linked again to the obesity phenotype. So it'd be interesting to be able to quantify these separately. If you have a look at these two mass spectra here, they actually show very unique ions, 57 and 74 for 2-methyl-butanoic, which is almost completely absent in 3-methyl-butanoic. Instead throw 60 and 43. And so if you extract those two EICs, you can see that even though there's almost no retention time difference between these 4 EICs, we can still separately quantify these 2 branched chain fatty acid isomers. So that's an equal bonus to the method that we've developed, which we're hoping will throw some light on the role that these play, again, in relation to the obesity phenotype and metabolic diseases involving chronic inflammation. The final bonus of this technique is all these other things that you can still see in the chromatogram, even on our slightly scaled back analysis. And these are an array of volatile compounds based around aldehydes. Some of them are male products, which could be artifactual, but are still possibly interesting to our analysis. And we've even managed to pull out some very low volume compounds of a few hits of trimethylene, which is another very important compound implication cardiac health and metabolic disease. So really impressed that we've managed to pick out something as small as that. Another target that we'd like to see is something like acetaldehyde. So, in conclusion, thermal desorption GCMS has excellent potential for discovery and analysis of volatile and semi-volatile components in the metabolome. We can provide both semi-quantitative and quantitative data and absolutely quantified data in a single analysis. Compared to other volatile sampling and sample introduction techniques, we have superior sensitivity, greatly enhanced diversity of applications, and further developing methods and workflows may come up some of the issues of fragility that we've encountered. So it's an exciting place to go. Thanks very much for listening. Can I take questions?
All right, thank you very much, Chris. Do you have any questions for Chris? Are there any questions? All right, no. All right, thank you very much, Chris.
Thank you for your patience.
Yeah, and thank you for the graphs. They really help us a lot to follow through everything. All right, so no. So now I just want to. Cool. I get to share. All right, so I will be doing the last talk. Just bear with me for a while. For those of you that don't know me, I'm Kyriakos. I'm one of Master's Research Fellow, and we are actually pimping me out. That means that you can hire me for your research purposes. We're not allowed to call me slave because it's kind of politically inappropriate and stuff, but you got the point. As Gas showed, you can go to resources, and we are offering two types of grants: research fellow support grants and instrument use grants. Usually they kind of go together. The instrument use grants mainly for people that have never used mass spectrometry, and we're just giving them $500 to do some initial runs. I'm going to talk a little bit more about the research fellow support grants. If you have a bunch of data and you don't know what to do about it, that's pretty much George. You just email George and then you can discuss. If you have a sample and you don't know what to do about it, most likely it's me, and that's where I jump in, and then it will be along with George. So I'm sharing my screen. Let's look a little bit about what we do on our proteomics lab. We can generally say that we can help you with some examples I'm going to say here. Let's say, for example, you have a whole tissue, either like cells or a whole tissue sample. We have lenses from eyes, we have tissue from heart or whatever it is you have. We can digest the whole tissue and see what's going on in the tissue. Something very, very, very interesting is that, as many people said before, when we have a significant amount of proteins, some proteins suppress the signal of other proteins, and you might not see proteins that are in a lower abundance. And if you want to see some proteins that are in lower abundance, you can actually do an SDS gel first, target the correct molecular weight, and then process and do proteomics. That will help you bring out the lower abundance proteins and give them signals a little bit of a boost. We can also confirm your findings of Western blotting, and we can also confirm if your immunoprecipitation was successful or not. The general step we follow is we reduce the disulfide bridges, we alkylate, and then we proceed to enzymatic digestion. I'm going to talk today about two main pathways we follow, but I will explain more later on with specific examples for expense as well, because most people are asking us how much does it cost, well, how can I set up, and how can I plan my project. After we have taken all of our data using our instruments, then we go to data analysis. On our website, we have uploaded a lot of softwares that we use. I'm just mainly focusing on these ones that they're giving you a lot of information, and they are the softwares that I mainly use. We have George that has an army of softwares. Protein Pilot usually is used to give us a library construction and general information of what's going on there and an idea of what's happening on the post-translational modification. We use Skyline, has better graphs. It can help us with the SWAT analysis and protein quantification. And it's very important that it can help us compare what we call most likely health and disease, or generally group A and group B. And then we have FragPipe. This is more specific for post-translation modifications like glycosylations and stuff. Now, let's take these two major different pathways. Let's say that you have a sample and you're just giving me a sample. You want to know what do you have in that sample. If it's just a dummy run, by the way, we don't charge. But if it's more runs, we will talk about later how much we charge. The discovery type, like the general library construction, is fast. It's very inexpensive. It gives us some preliminary data. It can help you write some funding application using that data, or it can confirm you findings of Western blottings. Physiologists swear like a Bible to their Western blottings. They are like, oh yes, that protein is there. And we're like, are you sure sunshine is there? So we can actually confirm it. What the Western blotting gave, it's actually true or not. Usually for a 30-minute run, it's 125, not usually, it's the 125 unless it's just a dummy run. Or if you want a better peptide fragmentation, we use 60-minute run, we have 175. These prices are for University of Auckland collaborators, not for commercial collaborators. Now, let's go to the second major pathway. I want to thank Michelle, Anastasia, and Olivia for their talks, because that means that I don't have to say anything. You already know what SWAT is, you already know what MRM is. Very quickly, we use this when we want to compare between two different groups. Say you have a healthy sample and disease sample, you have bacteria that are healthy, bacteria that are dying, you want to compare the different proteins, or anything that you can think in terms of comparison. We do need to say that this is a very good method either for standalone publications or to support your data that you already have. It's very good for funding applications that are at more demanding stages or huge funding applications like HRC. We advise 6 per each group. Otherwise, if you don't have a significant amount for each group, then it's not publishable. It's also not publishable. And as Olivia said, the MRM is very specific and it's very sensitive, and it will give you better peptides, minimizing and reducing the noise behind. In case that you want to do SWAT analysis, the first couple of steps is exactly the same. You will digest, you will process the steps like we do before. But the most important thing is that we obviously, when we're talking about quantification, you want to make sure that you are actually digesting for each sample the exact amount of protein. Otherwise, it doesn't make sense. So we are using EZQ technique to see how much protein we have in each sample, and then we are going very specific on how much protein we will be digesting for each sample. For this here, where we have to run just to see an overview and create a database, we don't charge. But then later on, we need to do what we call fractionation. If you remember from Anastasia's project, we have different fractionations here. We use different salt concentration. I do need to point out that the idea, what we say is that you cannot monopolize the research fellow. That means that we are using optimized conditions for other projects. For example, one of the projects was Anastasia's project. We spent a lot of time trying to optimize the best conditions. But you can play a lot. You don't have to do salt concentration. It may be pH, it may be different cytochromatographies, chromatographies. But I'm not willing to get another PhD. I already regret my first one. So we are going with what we have optimized. And if you want something else, then we have to discuss a little bit differently. Usually we go 10 to 14 different salt concentration. That will cost you approximately 2.5K for this process only. And then we obviously have to do our SWOT analysis. Thank you, Anastasia, for showing us exactly what the SWOT is. You get something like volcano plotting, heat maps. We have already discussed about this. That also costs, for our example that we said before, 6 control and 6 disease, 12 samples, and we usually do 60 minutes run, and that costs 2,000. Well, yeah, it depends on how many samples you have, obviously, but for this example. And then we will go usually on the MRM method. We give you the data, you choose your proteins of interest, and then we go back and target specific peptides because the MRM method is more sensitive. That's very cheap, $65 per sample. For the example that I gave, 12 samples, it will cost you $780. So for this project, it will cost you approximately 5K plus. The plan is that we will be doing the proteomics workshop. We did not cancel, we're just postponing it, hopefully somewhere in August. So do come along. And I would like to know also if you had a more, because I wanted to do a more hands-on workshop where we would show you the actual lab and the processes. If that would be something of interest to people, please do click me a message so that I can see how can that be organized. Do you have any questions on the previous one? So you can just, before you apply officially to MASH, you can contact myself or Martin or George, and we can help you and guide you through the proposal. And at this stage, I want to invite guys. Yeah. So we have Jacob, we have a small token of appreciation for you.
Yeah, we just wanted to officially present Jacob with a bottle of this toxic wine and an envelope of undisclosed, thing. To thank you very much for presenting your work this afternoon.
Like I said, happy to keep your captain. It's no worries.
Thank you very much.
Payment is liquor or
Yeah. And for one more time, I would like to thank our sponsors, MASH, Medical and Health Sciences, Morris Working Center, and Share Research Equipment for funding this event. One more round of applause for all of our speakers. Thank you very much.
Thanks for coming and for participating. And if you have more questions, just don't hesitate to email us. We're here to help you. MASH is a great platform to help you with your mass spec questions. So now it's time to enjoy that toxic wine or beer. Yeah, we gather in the tea room, FES tea room, and we can have some nibbles and drinks. Thank you.
Thank you very much.